giving your time for us to enlighten the things in deep learning.
Thank you very much, sir, for inviting me.
Yeah, thank you, sir. Thank you very much. I think in future also we may bother you. Please,
Sure.
please bear with
Yeah.
us.
Yeah.
Right, sir.
You're always welcome,
Yes,
sir.
thank you very much, sir. Nice to see you.
Yeah.
Nice to talk to you.
Yeah. Thank you very much for this time and the
Right, sir.
opportunity.
Right, sir. Right, sir.
Yeah.
Right, sir.
Yeah, very good morning to one and all. Hello.
Yes.
Yeah. I am Dr. A.
Good morning.
Sivashankari. Good morning, sir. I am Dr. A. Sivashankari, coordinator of this webinar. On behalf of entire GMRIT and Department of ECE, I feel privileged to welcome you all for this webinar on deep learning in biomedical image data analysis. Also, I extend my warm welcome to our beloved principal, Dr. CLV RSV Prasad, respected Vice Principal, Dr. J. Raja Murugadas, and our HOD of EC Department, Dr. M.V. Nasharaf, sir. Today, we have with us Dr. Ebenezer Daniel, Postdoctoral Fellow, City of Hope, National Medical Center, Cancer Research Institute, California, USA, as resource person. I extend my warm welcome, you, sir.
Thank you. Thank you very much.
Totally 685 participants have registered for this webinar. Great pleasure to welcome you all for the participants. Sir Daniel, he had completed his PhD in ECE from Karunya Institute of Technology and Science, Coimbatore. He had undergone his post-graduation in Karunya Institute of Technology and Science and undergraduation in Mahatma Gandhi University, Kerala. To add credibility in his research experience, he is a postdoctoral fellow in the Department of Diagnostic Radiology, City of Hope, National Medical Center, California. His research areas are structural MRI, diffusion MRI, machine learning, deep learning, PET-CT imaging, cancer diagnosis, autism identification, and aging research. He had published his research papers in various reputed indexed journals and conference publications. And he is also a journal reviewer in IEEE Signal Processing Letters, IEEE Sensor Journal, IEEE Publisher, Elsevier Publisher, and in many reputed journals. So once again, I welcome you all and have a good day. Now it's over to Dr. Ebenezer Daniel, sir, to take this session. Thank you.
Okay, thank you very much for your welcome address. Oh, can I start with my presentation right now?
Yes, sir. Proceed, sir. Proceed,
okay.
sir.
Yeah. So, can you see my PPT? Is it visible?
Not yet.
No, sir. No, sir.
Not yet. Not yet.
Yeah. I'll check once again.
Yeah, yes.
Oh, right now it's visible, right?
Yes, it is visible. Yes.
Oh, okay. Yeah. Good morning to all of you.
Yes.
Directly, I'm going to present my topic, deep learning in biomedical image data analysis. Specifically, I'm focusing on MRI brain image analysis. And thank you for this wonderful session and also inviting me to address this session. Myself, Ebenezer Daniel. Right now, I'm doing postdoctoral fellowship in Department of Diagnostic Radiology, City of Hope National Medical Center. It's a cancer research institute in California, USA. So my overall overview of presentation, the basic introduction of artificial intelligence, machine learning, deep learning, and very basic introduction to image processing, then deep learning network and the structural and function of brain, some neuroimaging data, case studies, some platforms, and then databases for medical image analysis and some job portal. Yeah. This is my research background. Already you introduced. I was a postdoctoral fellow in University of California, Los Angeles during 2018 to 2019. And I was mainly working on structural MRI, diffusion tensor MRI, and brain changes in heart failure patients. And since 2019 October, I'm a postdoctoral fellow in City of Hope Cancer Research Center, where I'm working on structural MRI, diffusion MRI, machine learning, deep learning, PET-CT imaging, specifically for bone marrow, then cancer diagnosis, autism, and aging research. So we all know about deep learning. Daily we hear about various aspects of deep learning in our day-to-day life. We know at present all the industries are welcoming deep learning and artificial intelligence ideas to change or reduce their like hand engineering, and they want to implement more automated nature. You know, various companies, various industries like well-known Facebook, Microsoft, Amazon, Google, IBM, Siemens, all, and various industries in automobile industry, social media, and medical imaging, and various places, they are changing into artificial intelligence and specifically into deep learning applications. So what is artificial intelligence? We all heard about artificial intelligence very well. Artificial intelligence is an ability of a computer program or a machine to think and learn. That is a basic definition for artificial intelligence. When we come to machine learning, it's a study of computer algorithm that improve automatically through experience, and it's a subset of AI. Machine learning is a subset of AI. When we come to deep learning, it's again subset of machine learning, where artificial neural basically follows the structure of artificial neural network. But we can use deep learning for more complex data, and some complex data we cannot directly apply in machine learning. So we can directly apply deep learning algorithms. And again, deep learning is a subset of machine learning. Deep learning also work like based on experience and training and learning procedure. So in deep learning, there are like more user-friendly or more automated. So this is the era of artificial intelligence. You can see from 1950 onwards, artificial intelligence is like started in our various industries and various applications. And after 1980s or mid of 1980s, the machine learning started into the picture. After 2000, here actually in 2006 or 2007, and very recently, the deep learning also started as a very advanced platform for artificial intelligence. So this is also like very common, like as I told in first slide, deep learning in everywhere, like image classification, object detection, speech recognition, or traffic, like a pedestrian detection or line detection, automated vehicles. And we usually discuss more in biomedical or medical imaging for diagnosis, treatment planning in breast cancer or whatever it may be, different cancer or degenerative diseases or various medical applications we using deep learning. So what is the basic difference between the machine learning and deep learning? Here, this picture, this is a very well-known picture. I think most of you have seen this picture already. Machine learning versus deep learning, the main difference between machine learning and deep learning. Here you can see the first picture. There is a car. Input image is a car. And how deep learning, how machine learning work, there is a feature extraction, then followed by classification and output. This is a car or not a car. It's a predicted. Whereas you can see the deep learning, there is an input. There is a deep learning platform. Togetherly, we call feature extraction and the classification, and it's predicted as a car. So in deep learning, in machine learning, you can see there is a more hand engineering is required. We need to select the features, we need to optimize the features, we need to find out what is a statistically significant features. So more hand engineering is required. Whereas in deep learning, we make it as more automated nature. So that is a very common or general difference between machine learning and deep learning. Here, deep learning in medical imaging application, as I told, the deep learning in various medical imaging applications like a slice detection, ligament isolation, classification, and various therapies like a treatment planning and all and various method-wise application, not only in diagnosis or treatment planning, whereas in various methods like artifacts removal, noise removal, where mostly the engineers like usually has interest on artifacts removal, noise removal, or reducing the slice like a plan sequence timing and all other automated applications we using deep learning in our medical imaging application. This is the you can see this is from PubMed, like a publication in medicine. So you can see the journals and papers, how deep learning is increasing in recent years. So this is the number of papers, number of publication, and here you can see the year-wise improvement. After 2014, 16, 18, you can see the very RAM changes in number of publications in deep learning. I know maybe most of you very familiar with the image processing, but I will give few slides on maybe some students who watching, they were like very first year or second year of their undergraduation program. They may be not much familiar with image processing. So I just give you few introduction slides on image processing. So image processing, these are the different platform. You can work on MATLAB, or you can work on any of Python platform, like or you can for your image processing. This is a very basic image. This is called a binary image. You can see the black and white. All the pixels are either black and or white. So you can. I'm selecting a small region. You can see the pixel values here: 1, 1, 0, 0, 0, 0, 1, 0, 0. This is like a very basic binary images. Here pixel values pixels either 1 or 0. So the image will appear either black or white. So we come to the other image called the grayscale image. Here the pixel value usually vary from 1 to 256. So you can see the picture 0 means it's a black and white means 255, and or we can get any gray scales, gray shade, white. The all regions supposed to be in the shade of black and white. So this is a grayscale images, and yeah, pixel close to 255 is a light pixels that become very bright, and 0 become very dark. So this close to 0 darker pixel represent the structures having structures or soft tissues usually. So basically this is a black and white. Sorry, this is a grayscale image. And when we come to color image, usually in color image we have 3 planes or multiple plane, like a red plane, green plane, and blue plane. Here is a combination of different gray scales, like a red plane, white, green plane, and blue plane. When we combine all this, we'll get a 3 plane or a color images. So you know, color ranges 0 to 255, and that gives total 255 power 3 different possible color combinations in color images. And since the total number of bits required for each pixel is 8. So together it will record 24-bit for color images. So this is just a different formats for saving, compressed formats for compressed or uncompressed formats for image saving, like BMP, TIFF, PNG, JPEG 2000, and these all like we knowingly or unknowingly we using in everyone in our day-to-day life for imaging. So when we come to brain imaging data, especially medical imaging data, we have different imaging like X-ray imaging, computer tomography or CT images, or we're all familiar with the MRI like a structural MRI, diffusion tensor imaging, functional MRI, positron emission tomography. There are a lot of other images which I am not going to address here, like ASL, perfusion imaging, and yeah, you all familiar with the different imaging, cerebral blood flow imaging, and a lot of other SPECT imaging, single photon emission, computer tomography, and different imaging modalities are available. So mostly I'm going to give you few like introduction about these 6 imaging modalities. So this is just a brain structure and function. You maybe think it may be like a little bit awkward. Yeah, so I want to say when we are doing imaging, we have to find different things, like you can see the skull and different lobes like parietal, occipital, cerebellum, frontal lobe, temporal lobe, brain stem. And here I have just given, you can search in Google, you will find out different lobes, what are the functions. So when we are doing imaging, so these all things are brain functions also very important. This is a very basic imaging called X-ray imaging. You can see how X-ray generator, and there is an X-ray generator, there is an X-ray detector based, and in between we place our object. So X-ray can penetrate through liquids, gas, and solids. Bones, which are heavier atom than flesh, absorb the X-rays. So you can see that usually X-rays work like a negative imaging. So based on the negative imaging, the radiograph, we will get a radiograph, and mostly we can identify this bone images using X-ray imaging. So there is another very common imaging modality called computer tomography imaging. So here I'm just explaining few hints of imaging modalities, and I will go to the deep learning. So this is a computer tomography imaging. Computer tomography is basically working based on the principle of X-ray imaging, but compared to conventional X-rays, here you can see there is a rotating X-rays. So we can find out not only one direction, we can get a multiple direction pictures or a series of images in using CT imaging. So here they're taking a CT of brain, CT of any other organ. So this is more advanced form than conventional X-ray imaging. So other one very common imaging like magnetic resonance imaging. So magnetic resonance imaging, there is a strong magnetic field, radio frequency coil, gradient coils. So we used to place the patient in between two magnetic field, and we apply a magnetic field, and we need to find out based on the magnetic field, we can find out the changes in our body, and we can make it as an image. So there are a lot of research in RF design, pulse sequence optimization, imaging, also what kind of gradient we are using in MRI imaging. So this is a very basic picture of MRI imaging, and it's not much important, but you will get some idea about how MRI is working. So the very first imaging is structural MRI. Structural MRI means the imaging, we will get more structure portion or more soft tissue portions. So structural MRI we usually call SMRI, which represents structural MRI. This maybe we are using for tumor identification or any kind of abnormalities in brain. There are different kind of structural MRIs, T1 weighted images, T2 weighted images, and the flare. You can see here T1 weighted, T2 weighted, flare, and different. These all are comes under the structural imaging category. So in T1 weighted imaging, you can see the CSF. This is the CSF, sorry, cerebrospinal fluid. The CSF is black, and if you go to the T2 imaging, CSF is more bright, and if you go to the flare, there are fluid attenuated inversion recovery. So here you can see the fluid region also. The fluid also little bit black or shady. Here its fluid is bright. And in flare imaging, there is no fluid. We can avoid. We can attenuate the fluid. So because sometimes if there is a fluid, we cannot diagnose the like tumor or the abnormalities properly. So we can, this is also an advanced form of imaging. So any of these images like a T1 or T2 flare, we can represent in different views. Like we can represent in axial view, we can represent in sagittal view, we can represent in coronal view. These are the some basics of structural MRI. And structural MRI is giving mostly the data in micrometer unit, whereas this will give in very deep tissue information like a diffusion tensor imaging. Diffusion tensor imaging, we will get the very narrow down minute details like white matter, like a tissue integrity in our brain. So this is also very advanced imaging technology, which is usually called DTI. So this DTI imaging, we will get the axonal connectivity in our brain. We know the axon myelin in our neurons. So the diffusion tensor imaging is using for the axonal connectivity in our brain. So the another one imaging formal techniques called functional imaging. So the functional imaging, you can see there is two pictures. From this picture, you may easily understand. So this is for a stroke patient. So for the stroke patient, this first showing is a healthy subject, and below it showing is a stroke patient. So you can see the various activation changes in the brain, how a stroke patient's brain, while he is moving his hand, how the brain activation is changing for a healthy control and for a stroke patient. So this is called a functional MRI imaging. Basic principle behind the functional MRI is like oxygen-rich blood responds to the magnetic field of MRI differently than oxygen depleted blood, how it's responding. This is also one advanced imaging. So here we can mostly identify the function of each and every brain region in different patients. This is another. So most of this MRI imaging, whichever I have given here, we can use without any contrast. So we without any contrast means that is not much harmful. Ideally speaking, there is not harmful. When we using some contrast agent, then only it become harmful. It is less harmful than CT, X-ray, or even PET, positron emission tomography. This is another imaging techniques called positron emission tomography. This is usually called metabolic imaging. So here we can get the metabolic nature activities in our brain. So this is there is a healthy subject, and this is a Alzheimer's patient. So you can see how the brain changing in healthy and Alzheimer's while doing positron emission tomography. This is usually for treatment and planning, surgery, and of various clinical applications. Here, there is a radioactive or traces have been absorbed into the bloodstream. Based on that, we can identify the activation. Like the principle behind is active areas of the brain will be utilizing glucose higher than inactive areas. So based on that principle, we can on inactive regions in a brain. This is also very commonly using imaging. So usually this PET imaging and all, it's called metabolic imaging. So the metabolic imaging, there is not much structural orientation. It's mostly giving the metabolic activities or a functional activities of brain. So we need to, when we come to the picture, we need to merge these images into some structural images. So this is, you can see This is an MRI of a whole body skeletal. So here this is a PET, and this third one, C, is the fused images. So fused images, we can identify exactly, we can locate where is the metabolic activity and how it is important. So similarly, this is a CT. CT also is a very hard bone structure. It's also structural images and corresponding PET images, and this is a fused image for CT PET images. So we have seen already different imaging formats like JPEG, PNG, TIFF, and all for our natural or imaging. But whereas in all these medical images, we used to keep in a formats called DICOM, usually digital imaging and communication in medicine. That's a different standards, or DCM. This developed in 90s, around 1993 onwards, it become very standard in medical imaging. And there is another recently developed imaging standard called NIFTY or .NII, Neuroimaging Informatics Technology Institute, by National Institute of Health of United States. And most of the brain imaging presently people working. There are many other advanced platforms, but most commonly people, or most widely, they are using with the NIFTY format. So we'll come to the deep learning network. Deep learning, we already seen the difference between the conventional machine learning approach and the deep learning approach. So before going to the deep learning, I just explained various imaging modalities in medical imaging. Deep learning network is an artificial neural network with multiple layers between input and output layer. You can see this is an input layer. So input layer which connected with the input images, and this is an output layer, so which is connected to the output classes. In between, there are some layers which is called hidden layers. Here, this you can see the hidden layer 1, hidden layer 2, hidden layer 3. Like depends upon our application. We had a number of hidden layers. So, in basically speaking, in deep learning network, using conventional neural network approach, we have input layers and hidden layers and output layers. So the layers in between input and output is called hidden layers, and in deep learning, these all layers, hidden layers, are very fully connected or densely connected. So I think, just a minute. Oh yeah, I'm sorry, I missed one slide somehow. Yeah. So the deep learning, the hidden layer unit is with different blocks. The first block in hidden layer unit is a convolutional block. So this convolutional block will generate some feature maps, and it works on the basic principle of convolution. Here you can see one input image. There is a convolutional kernel, and which generate a feature map. So this is a convolution operation. So this is the very common building block in hidden layer. So you can use the convolutional kernel in different size. Here you can see we were taken 3x3 convolutional layer. So, and another one important layer in hidden layer is is an activation unit. There are different activation functions. You may heard about sigmoid activation and various other activations, but here I would like to give most commonly using or most standardized activation unit called rectified linear unit, or ReLU. So ReLU is like our electronics half-wave rectifier. It's most likely like our half-wave rectifier. When the negative inputs, it will give 0, and when it is positive, it will linearly increase. So this is an activation unit. So this is called one activation unit, usually connected with our convolution layer. Another block in our hidden layer. Hidden layer, already we have seen convolution, and already we have seen the other layer called activation, and another one is called pooling layer. So we usually use the maximum pooling. Here in maximum pooling, you can see this is a 2x2 maximum pooling. So it will divide into 2x2. Yeah, here this is the first 2x2. What is the maximum value? 6. And then another 2x2 where the maximum value is 8. Another 2 cross 2, the maximum value is 3, and again another 2 cross 2, maximum value is 4. So likewise, we can reduce the dimensionality of the input images, and we can make it into a single array. So another most commonly using layer in hidden layers, some cases like even in VGG networks, VGG 16, and they were not using dropout layers in hidden layers, but sometimes when we use with the more complex data, we have to use dropout layers. That depends upon our application. Dropout layers can be used in hidden layers as well as in output layers. The main why we are using dropout layers, we have to avoid the overfitting. The overfitting is nothing but the network is started to memorizing rather than learning. So we don't want to get the memorized output. When we memorize the output, it will give, while it's training, it will give some results which is very good accuracy or less error. But when you go to the practical testing scenario, it won't give the accurate results. So we need to avoid the overfitting. So we used to use the dropout layers. So you can see this is a standard neural network, and this is that applying after dropout. So we can avoid all the dead nodes in dropout layers. This is another. These are the main blocks in hidden layers. Once we come to the end of hidden layer, there is called a flattening layer. So you can see the flattening layer. This 2 cross 2 matrix is converted into a single column matrix. So this is called flattening. So here we have taken only 3 cross 3 and made it as a single column. So another one activation function is called sigmoid activation. Sigmoid or softmax we usually use for the output class prediction based on our application, based on number of output classes. So this is a very basic sigmoid activation, and we use for mostly for sigmoid. Mostly the researchers that very recent papers which showing people using the sigmoid activation in output layer for output class prediction rather than using in hidden layers, because when we using ReLU in hidden layers, the our training efficiency is high. So when we going for implementing this deep learning, very common platform like a TensorFlow plus with the Keras with the TensorFlow background. So this is a very TensorFlow is developed by Google. Yeah, this is a very common deep learning platform. Here I would like to give you few sessions or few lines. So this is a MRI images, just one slice of MRI image. I have taken only one slice of MRI images. So with dimension 128 cross 128. So first of all, this is a hidden layer or deep learning designing. So we need to give a sequential model. Then our hidden layer is with 128 cross 128. So we use some convolutional filter with 32 convolutional filter, then with a kernel size of 3 cross 3 with the activation of ReLU. Then we will go for the maximum pooling of 2 cross 2, then dropout of 0.25. 0.25 means 25% of dead nodes we are replacing. That's totally up to you, because sometimes you don't need to drop out layers. Here we can use batch normalization, dropout layer. The depends upon those layers, the complexity may be increased. So you can avoid or you can include or exclude, depends upon your nature. So this scenario you can see the input images is 128 cross 128. After we are using 32 filters, so the output we will get a 32 feature maps, and the output feature map size, again we will go to the next session. The input image size is reducing because here we are using the maximum pooling. The input image size is reducing to half. So the second hidden layer, the input image is size with the size is 64 cross 64. So definitely we are increasing the number of filters, because this is not standard for format 32, 64, 128. You can use here 256 or 128 or any number. You may be very familiar, because why we use this VGG kind of architecture means, when we reduce the features and the input image size is like very high in some cases of medical imaging, like ImageNet or GoogleNet or some other images, they may be using 64 cross 64 or sometimes 32 cross 32 size. But in medical imaging, we usually work with minimum 256 cross 256 or 220 or nearly that. So for us, the each and every pixel is very important. So that time, when we apply 256 filters at first time, the complexity may be very high. So we can select less number of filters with small kernel size at the first hidden layer unit, and eventually we can increase the number of filters like a 64. And so of course the size of the image is also reducing in next level after maximum pooling. So here you can see that third hidden layer, like 128 filters using. So hidden layer. So after the third unit, it will be reducing to 16 cross 16. So the output layer, usually with the flattened layer, densely connected layers, and activation layer. So here you can see using this command this is all based on TensorFlow. So you can add flattened layer, which convert all 3D feature maps into 1D feature vector. Then you can add the dense layer. You can add the rectified linear unit activation. You can add dropout layer. Then finally you can give again a dense layer and final activation unit for prediction. Then you can compile your model. Also you can train your model, like you can decide what is the batch size you want. This is this XY is the X image and Y is the label. Like what is the batch size you want, and what is the validation split, like your total images. Do you want to give the 0.3 validation split, means 30% of data we are using for the validation and remaining 70 for training. So how many purchase record, how many times you want to pass all the data. So when you give more repurchase, usually you will get more trained and more good validation. So roughly speaking, you can take 50, 100 or different number of depends upon your application, depends upon your number of input images. And when you use more repurchase, of course your training time will be increasing, and you can save your model with any name. And if you in future you can use that model like a transfer learning and other like a different applications you will be familiar. Once you can save your model and further you can use. So this is what all we explained so far. So the input layer, and there is an output layer. In between layers called hidden layers, and hidden layers is filled with like a convolution layer, activation function, maximum pooling, and dropout layers. So here, this is the programming what we have seen already. Hidden layer 1 with the convolution layer 32 activation, ReLU, and maximum pooling 2 cross 2 and dropout of 50%. And second hidden layer with convolution 64 and activation, ReLU, and maximum pooling 2 cross 2 and dropout. Similarly, in output layer with the flattened layer, dense layer 1, dense layer 2, and activation with the sigmoid activation. So this is just a general model where I am using only 2 output classes. So I use the sigmoid activation. If you have more, you can take another activation functions. So other deep learning platforms, like so here we used TensorFlow with Keras, and like a PyTorch, DLTK. The TensorFlow is designed and developed by Google, and PyTorch with Facebook, and DLTK is specifically for deep learning toolkit. And this is very new deep learning platforms. So this is called NiftyNet. NiftyNet is an open source based on TensorFlow. This is very recently developed, I think 2017 or nearly, by Imperial College London, their medical imaging research team. So this is like specifically for 3D Nifty images. So you can work the medical images directly in NiftyNet. You don't need to bother to convert into 2D or selecting the maximum informative slices and all. You can use this NiftyNet. NiftyNet modular structure is designed for sharing network, and you can use it for a pre-trained models. Yeah, this is a very new tool, and the features like you can easy for interfacing, and you can use for 2D to 3D, 4D input images. You can use for UNet, VNet, and various applications like segmentation and prediction. In various applications, you can use the NiftyNet. Other very recent, I believe this is very recent, and as best of my knowledge, this is developed by University of Southern California, UOC research team. This is called a Nifty Torch. This is an advanced platform like a PyTorch for the medical imaging, Nifty imaging applications. This is a PyTorch embedded pipeline, not like based on TensorFlow. It's mostly based on the PyTorch. So yeah, this is also very new platform. You can just search and you can download this Nifty Torch also. There are many other like as we discussed, the NiftyNet, DLTK, DeepMedic, UNet, for especially for biomedical image. If you're working on image segmentation applications, you can use VNet, UNet, then VNet, SegNet, and various deep MRI reconstruction platforms. There are a lot of other platforms. So just like I would like to give you a few hints on some few hints on some case studies in deep learning, like ASI identification, autism spectrum identification, lung cancer, brain metastasis, aging study, cancer identification, and for some degenerative diseases. So again, I am giving you one basic difference. Already we have seen the basic difference between machine learning and deep learning in imaging applications. So this is the one tumor identification platform for network for using machine learning. So this is the structural images. So when we need to do segmentation, then we have to extract the features. All the features we have to extract. Then we need to go for the statistically significant feature selection. Then we have to give for the classification unit. Finally, we will get the classified unit. This is a conventional machine learning approach. Whereas the similar way we can use, we have different input images, and we can train a network so that without any manual feature selection, we can train our hidden layers, and we can get the predicted output classes. This is what we were discussing already. So this is one case study. Before going to case study, I have just given an overview. Deep learning in ASI. ASI means autism spectrum identification. So the objective, I'm just giving a case study. The objective of present study is to develop an efficient deep learning network to diagnose the autism spectrum using structural MRI brain. And about 1 in 59 patients is identified with the autism spectrum density. So this is a case study where we are diagnosing the autism spectrum from gray matter density. So before going to the identification, and I want to give you the structural brain changes or gray matter changes in autism ASD patients. You can see the various brain regions, how this is changing. Middle temporal gyrus, you can see the changes. These all are the reduced gray matter density here in fusiform gyrus and various brain region of interest. So the ASD or autism patients, so their brain gray matter density is reducing or decreasing compared to a healthy control, age matched a healthy control. So this is we know this scenario. So based on without comparative analysis, based on the gray matter density maps, how we can automatically classify or identify the network. So very basically, sometimes we can use, this is a whole brain, you can see the whole brain. Most of the time we can use the whole brain or any organ, especially whole brain. Specifically speaking, deep learning is more automated, so we don't need feature level extraction like a hand engineering, like a segmentation approaches for extracting some features, but sometimes we need a pre-processing steps. So the pre-processing steps, which is our deep learning procedure, because the whole brain, you have seen, this is from the gray matter itself. Brain, like various various tissues like a gray matter, white matter, CSF, skull area, and various brain areas. So we don't need to train, or we don't need to do the convolution operation and all the deep learning steps on all the brain regions, because from the gray matter itself, the previous studies shows that we can easily differentiate the brain in an autism patients. So for that, we were just segmenting the gray matter. So this is a procedure we use as software SPM. This is very open software. You maybe heard about this SPM 12 version. You can use SPM 12, SPM 8, different SPM versions are available. So using the segment option here, we can segment the gray matter. Then we will normalize into MNI space. So we will convert into a standard space. Then we will using a smoothing operation, and we will get a smooth, normalized smooth gray matter. So based on, instead of this whole brain, here in the study we use the, instead of this whole brain, in the study we use the normalized gray matter. So however, you can use the whole brain, you can use the smooth gray map. So when you use the as according to my knowledge, or as best of my understanding, rather than using the whole brain, you can go for a specific brain tissue that give you more accurate and less complex identification. So this is a network. So here your input image is 256 cross 256. The 70 means there are a lot of slices, like NMR images, which consists of nearly 200 or 200 plus slices, and which we have selected 70 slices, which are the more relevant or more informative, and the slices which consists of more tissue regions. So we have selected that slices. Then that is an input unit. That is not our deep learning procedure. Then our main idea, our main agenda, our main target is to train that hidden layer unit. So this is a hidden layer unit. Here we used 256 cross 256 as the input, but in the conventional, as I mentioned, many conventional ImageNet or ImageNet challenges or whatever it may be, Google Net, they most of the time, but I'm not much familiar with the conventional imaging, but as of my understanding, they use sometimes 32 cross 32, 64, 128. But here in medical imaging, though we didn't use our whole brain, we use the full size, like a 256 cross 256. So this all stages, like this is, I'm not repeating again, all like which follows the same standards of our previous architecture, like a code which I have shown you. Input image size, then we use for convolution, 32 convolution filters, activation, then maximum pooling, here dropout of 50%. So this dropout is designed based on our trial and error method, because when we were not using the dropout, there is a chance of getting overfitting, and the system, our network is started to more memorizing. So this is again, after we know the maximum pooling, this 256 become 128. So this is the second hidden layer unit, where the input images size is reduced from 256 to 128. Number of slices remaining same 70. Here we sequentially we started to increase the number of filters. Basically, this is the VGG architecture format, because in VGG architecture format, the number of the kernel size is small, like a 3x3, and where you can see the number of filters are sequentially increasing, whereas the image size is dimensionality is reducing also. So here you can see the 128 cross 128, where 70 slices with the 64 filters. So all of the remaining stages, you can sometimes you can skip, maybe drop out into different, or you can skip some unit. But I used every hidden layers uniformly. So this is next. Again, you're going to the hidden layer 3, 64 cross 70, 64 128. Convolution filters increased sequentially like 128. Then here, when you come here, this is the next stage, hidden layer 4. Input dimensionality is again reduced to 32 cross 32, but we sequentially increase the number of filters into 256. Then again, next stage, after 256, we can go for vital, but again, next hidden layer unit, I use the same number of like filters, 256. Here 16 cross 16. That's our choice, totally our choice. Then I went to next hidden layer unit. Again, dimensionality is reduced to 8 cross. Finally, from 256, I reduced to 8 cross 8, and with vital feature maps. Then I give to the output layer unit. There is a flatten layers, dense layers, and activation of sigmoid activation, and finally the predicting like autism or healthy control. This is just an overview of a case study I have shown you. So then this is not an accurate result. This result is just for the understanding I'm showing you, because the accurate paper is I cannot show you right now the accurate results for some other reasons. So we can show the loss function. Also, we can find out the loss. So of course, the loss supposed to, or error rate supposed to be less, and we have to get the accuracy. The different colors which indicate, the blue indicate the training loss, and the other color orange which showing the validation or testing. Similarly, in here it's accuracy, how it is increasing. Here you can see that in each, for various e-purchase 100, you can see the difference between, for the e-purchase 20, after e-purchase 20 is almost steady, but even though the difference between the training and validation error and the difference between the training and validation accuracy is very minimal. If you see the huge difference in training and validation, that maybe look like kind of overfitting, but here you can see there is no overfitting. The difference between the validation and training is very minimal. Yeah, then I would like to show you one research work. This is probably, this is mostly with the machine learning approach, not with the deep learning approach. This is a radiologic prediction of mutation status based on MR imaging of lung cancer brain metastasis. This is our paper. Recently, we have published one research paper from our research group. So this is the case where the lung cancer patients, they shows brain metastasis. So and we worked for the prediction of brain metastasis using the structural T1 and T2 images. This is a conventional machine learning approach, segmenting and selecting the different features, and finally we are predicting our type of mutation like EGFR or ALK or KRAS mutation, what kind of mutation belongs to the each brain metastasis. So that was the one of our study. This is just for the understanding I have shown you. There are other studies like aging study. So this is also very like a good study, brain aging study using machine learning. So you can specifically focus on some patient group, or specifically you can for a general healthy control, how their brain aging is happening and how it is related to chronological aging. So this is also another kind of important study for the deep learning application. This is also this is another study for cancer imaging. You can see the PET CT imaging. So this is focused on machine learning approach. So you can segment this is a PET, this is CT. You can use any software. Here we are using a software called LifeX software. You can use any software for segmenting or using the threshold based segmentation. Here using VOE segmentation, volume of interest segmentation. So once you get the VOE, then you can select the VOE features using Pyrodynamics or LifeX software. Any software you can use various features like intensity, log, shape, texture, wavelet. Then you can select the model. You can you do the correlation analysis. You can fix the optimal features. Then you can give for the prediction by SVM, random forest, or CNN, any kind of classification network. Other studies, just I'm not going to much details, but I'm just giving some hints, like various other studies based on brain MRI, brain imaging in biomedical imaging, cancer imaging, diabetes, Parkinson's, Alzheimer's, MCI, like various studies are presently going on in the field of deep learning based on medical imaging. You can, the basic block diagram seems to be mostly same. You can go much detail with the various research papers. So these are some pre-processing. Like when we go to some specific analysis, especially some based on some function, we can use the SPM. This is all freely available SPM, which works on MATLAB and C, VBM, FreeCell for FSL, Nifty segmentation, ITK, Elastic, DTI, TK. These all are the different pre-processing toolboxes you can use. So this is the pre-processing. So here there are different pre-processing. This pre-processing is not part of deep learning. I'm not speaking this pre-processing is not part of deep learning, but when you are feeding to deep learning, you can make your input layer become more accurate for the training or feature, getting the more accurate features. So this is the process of pre-processing. What I meant just to the SPM. So you from whole brain you can get a gray matter, white matter, CSF, skull, scalp, other tissues. So usually our brain activation, our regions with our patient groups is associated with either gray matter, or if you are using structural T1 or T2, can focus on T1 image, especially in T1 images, you can focus on gray matter. Focusing on DTI, you can use on the white matter, or if you're using T2, you can use both gray and white. So those cases, CSF is not important. So if CSF and other scalp, other tissues is not important, why we need to take these all regions, and why we need to make it as more complex? So the unwanted regions we can segment and we can eliminate using any of the pre-processing procedures. So when we come to our databases, mostly you may ask how to get these databases. So there are a lot of databases. I'm just showing some databases. This is the ADNI databases, Alzheimer's Dementia Neuro Imaging Interface. So this is the database. Here you can see DMRI, that is a diffusion MRI, fMRI, functional MRI, structural MRI, PET imaging, lot of images. This is an open source. You can access it from anywhere. 3,195 data is available in this single ADNI itself. You can go here, ABID. This is an ABID database. People usually working mostly on ABID. ABID is called Autism Brain Imaging Data Exchange. So this is an autism database. You can see 1,112 data is available in this ABID, mostly functional and structural MRI. T1 images, and resting state bold fMRI images. So similarly, you can see here at D 131 images, Alzheimer's dementia neuro interface. This is for depression, 131 images. So there are a lot of other imaging. Yeah, here you can see the aging, then different some Parkinson's database. See, this is a PPMI for Parkinson's disease, 2,298 freely available images. This is a human connect on recent project, 40 structural MRI for aging study. You can use like different imaging. This all belongs to Lonnie Group of University of Southern California. Not only this, you can get various open images, especially in brain images. All part you can get it, but specifically I'm speaking about brain imaging. You can get it from Cambridge CAN aging data set or Imperial College London. They are also giving free databases for mostly people are working on those databases, number of data images from the databases. Some of the databases, they may ask you the details like what kind of research plan and what kind of research you are going to do on with the data and who is the PI and yeah, what is your expected outputs and all. That's not so like very big steps. You can give your overview and plans so you can access the data very free of course. And this is a society called Medical Image Computing and Computer Assisted Invention Society, MICAI. So you can find out different job positions like a faculty job, postdoc jobs, research assistant, research engineer. And MICAI usually they conduct every year one conference. And right now, I didn't go for that. Another, you may heard about MIDL, Medical Imaging in Deep Learning Community. They conducted 2 conferences, and this year they are going to conduct 3rd conference in online. So there are a lot of other communities or also in medical imaging. Yeah, this is all about my presentation. I think I reached the time. Thank you very much for listening me, and thank you very much especially for the college and Dr. Arun and the head of the department and principal and all the members, especially all who attended, everyone. Thank you very much. If you have any question, you can share. I think Arun will forward to me.
Hello.
Hi.
Sir, we have received some questions from the participants, sir. Let me read the question. Can we give input to the EEG signal as image into the deep learning model or not?
Sorry, once again the question.
Can we give input to the EEG signal as image into the deep learning model or not?
Yeah, we can use EEG signal. EEG signal also we can use for deep learning applications. Yeah, but here I am specifically concentrating on imaging data. Yes, I'm not much familiar, but many people working on EEG in machine learning and deep learning applications. We can use that, but depends upon the application for what kind of like epilepsy or what is their study, based on that they can use.
Sir, next question. How CNN works on these structural images? Please explain.
Yeah, CNN. Yeah, let me go to the. Yeah, structural images are nothing but they give the soft tissue information, and structural images which consists of gray matter, white matter, brain tissues. So if you are specifically working on the same way how you are training other networks. See, this is all examples are given from structural images only, T1 images only. Same way you can use the T2 images, any other kind of structural images. Mainly when we go with the structural images or functional images, the problem with the pre-processing of the data. So we have to use the proper pre-processing toolboxes to eliminate or the unwanted regions, or normalizing the images into all the images into a standard space. Then because maybe the images occurred in different person or different perspective, because our number of cases may be very high, so that time we have to normalize all the images into same standard form. Like normalization includes registration, rotation, and making into a standard space. Usually we call MNI space. Yeah, or I used to work taking everything into MNI space. So mostly people combine into MNI space. Once you normalized into MNI space, then you can work like the same way, whatever I have shown in the code, or whatever the standard CNN form we can work. Yeah. Next question.
What is the basic difference in the various formats in your field?
Sorry, I didn't hear.
What is the basic difference in the various data formats in biological field?
Yeah, there are different formats. Yeah, I have shown DICOM and Nifty, which is more associated with our medical imaging. Most of the medical images, they use the DICOM images, DICOM format for the data storage, transmission, protection, and this is a kind of lossless compression or lossless data format. So specifically speaking, DICOM is the most medical imaging or biomedical imaging format. And when I narrow down into brain imaging, we use Nifty format. Yeah, mostly DICOM, and there are different formats, but this all basically I'm speaking about brain imaging perspective.
Next, how can we reduce or eliminate overfitting?
How to reduce or overfitting, right? Yeah, that's what I was discussing in this. Yeah, this is eliminating the overfitting by the very basic, like when we use more number of hidden layers. When we use more number of hidden layers, of course, here you can see 3 hidden layers. When we use more number of hidden layers, the chance of getting overfitting is very high, because when we use more layers, more number of hidden layers, more hidden layer units like convolution and all the batch normalization and all, the chance of getting what is called overfitting is high. So basically we can give an optimal hidden layer design, like based on our input image size, based on number of images, based on our output classes. We have to design our hidden layers in an optimal way. That is also one way to reduce the overfitting. And answer to the question, specifically we can reduce by using our dropout layers. Also, dropout layer is also very important in reducing the overfitting. Yeah. Any other?
How many research articles are published? Yes, sir.
Yeah,
how many research articles are published using this Nifty net?
Oh, Nifty net based publication. Exactly, I don't know, but I think Nifty net is a citation something like a 57 or nearly 100. When I was checking last, it reached something 60, 70 publication citation for Nifty net. Yeah, this is developed in 2017. So people were working in after 2017. You can say people, research groups are working after 2017. There are other platforms also developing parallelly, but many people are working in Nifty net. I don't know the exact count. You can see that I didn't check the reason how many publications in Nifty net. Yeah. Nifty Torch is very new as of my understanding, because recently I have seen the post in LinkedIn from that Loni group. That's what I followed, the Nifty Torch. Yeah.
Which is the best deep learning technique used for medical image segmentation?
Oh yeah, of course. As of best of my knowledge, I will say the deep learning network is UNet. UNet is the best technique for segmentation applications in biomedical imaging. That is that specific network. You can use the trained UNet, or you can make it your own way for training the network. This is the best of my understanding. UNet is the very good platform for image segmentation in biomedical images.
Brief about research scope for autism detection.
Yeah, artifact reduction. Yeah, I'll tell you. Many people working in artifact reduction. Artifacts usually occurring. You know, sometimes if you have some patients, they are mentally ill or facing some challenges, like, or some kids, like very young kids, they won't able to understand our instruction, like very small kids, you know. When we placing the small kids or the patients who's facing some challenges, they may move their head or any organ, even if not only in head. By my understanding, I work with the MRI brain teams, so they slightly move their brain. So we used to go, if they are able to understand, we will go to the scanner room and we will tell, please be steady for some time, steady for some time. But you know, this is a long time scanning procedures. Sometimes they change their head, change their body, they move. So this movement in their body make artifacts. So that scope of doing artifact removal is very good. So still, when we getting artifacts, our entire results will get like mostly affected, and we won't get the accurate results. So we have to use some automated deep learning based artifacts removal techniques and making into a standard like a normal space format. That will be very good scope for artifacts removal. Usually the motion artifacts is the main reason. Sometimes we cannot do remove the artifact, so that time we have to eliminate that slice images, or we have to redo the scanning. There is no other way. But yeah, some pediatric very small kids and all, we cannot do multiple times. So we have, if there is some automated algorithm, that will be very useful. Yeah.
Is it possible to detect brain metastasis from breast cancer with blood images?
From blood breast
cancer.
I don't know about that. This is like usually our patients, lung cancer patients, they have some kind of tumors in their brain, and the recent studies are mainly going on with like what are the cause of this tumors, and what are that mutation behind, like what kind of mutation, CRAS or ALK, EGFR, or what kind of mutation, and identifying the mutation and studying about the different cause and symptoms, and how this, and the location of brain, where is the where is this like exactly this tumors occurring and all mainly. But I'm not much sure about that. Is an entirely different research area, blood images and how to identify. Yeah, that may be good area, but we need more literature study. Somebody has to do in that blood images, and maybe they can develop in new way. Yeah.
How to use CNN for breast cancer detection using MATLAB?
Oh yeah, that is a lot of people working in that area. Not for, it's for detection, right? Yeah, that we can use it for X-ray, CT, MRI, all kind of breast images. They can. That's a very wide scope. We can use it for. But when you asking me about the deep learning in MATLAB, I don't recommend that much, because if you're using deep learning platform in MATLAB toolbox, that is quite expensive, and people, you can take the images or you can do the pre-processing steps in MATLAB, but once you pre-processed, you go to the any of the uh, platform like Google Colab. Like recently, many of our students also working from India in using Google Colab, and even like when we are doing work from home, we don't have much access to our server. Then we can use the Google Colab or like a cloud platform. So you can use the GPU also from your home, from any of your system. So I recommend use any of the Python platform like Jupyter Notebook or Google Colab or Spider or R or something else, rather working on MATLAB. These all are very user friendly. These are, you have a lot of help. It's available in online. Then you are working in MATLAB. When we think MATLAB is very helpful, I'm not saying no to MATLAB, but when we say about the more user friendly, you can work and you can get a lot of I don't remember the breast images databases, but a lot of good databases also available for X-ray and MRI breast images. Yeah.
Why we have to place 70 in between two 56s input layer?
Oh, that's a number of slices. Like this is not a single slice image. Usually in our, we'll go to there. Yeah, that's a very good question. Oh yeah, this is our usually we capturing an image. That means like a two-dimensional image with a row and column. But here it's a series of images, slices, vertical, horizontal, not like a one image. So your MRI, you have maybe 170 images, 216 images, or number of images. Among those images, this is like a movie. It's like a moving images. So among these images, we are selecting 70 best slices for each patient. So each patient image consists of 70 slices. That is what we mentioned. Usually the DICOM images and all nifty DICOM all images, not one slice. It's a series of images, all the direction images. Yeah, that's what 70. That 70 stands for number of slices. Yeah, any you can use, but commonly we use 70. You can increase to 100. You can reduce to 60 or 50. That's totally fine.
EEG is a data point, but converting them as images will make a confusion.
Age, right?
EEG
EG
is a data point, but converting them as images will make a confusion.
Oh yeah, but frankly speaking, I didn't work on much on EEG, but I have only vague idea about EEG, so I don't want to make my comments on EEG signal processing. But my understanding, people can convert into the corresponding imaging model. That's what my understanding, but I didn't work. I'm really sorry for that. I didn't work on EEG signal processing, so it's not good to comment on EEG. But as of my understanding, you can work. You can search on EEG lab or SPM, not SPM, some other EEG lab or some other softwares, and you can just check it. Yeah, what are the networks available? What are the connection networks available for EEG and all? Yeah, brain connection networks. I don't have much idea about EEG.
For breast cancer, which how can we find breast cancer?
Can you
And will
please?
suggest ultrasound days.
Sorry, I didn't hear at all. Can you just repeat the question?
For breast cancer,
Mm-hmm.
which algorithm is best and how can we find the breast cancer?
Oh yeah, the algorithm means I don't know what is the exactly question I asked, but my understanding, you can you have to find the changes, like mostly the breast cancer, like the early stage identification, and really can concentrate in that way, like how to identify very early stages using your imaging technology, or lot of data analyzing in a single time, or more accurate data analysis, and you can do that all segmentation and thresholding, all conventional approaches, and you can train your network. Yeah, specifically there is not single algorithm, there are a lot of algorithms, there are a lot of approaches. You can concentrate on some early detection, you can more accurate detection, more efficient detection, processing more number of data. Yeah, that and all very good way in those researches.
Conversion of unstructured to structured data, how it can be done?
That question is not clear. Unstructured to what they mean by structure? It's a image structure or some computer science format of structure. I don't know what is the question exactly mean. Data formats, or they mean the structural and functional structure.
Okay, so we'll move to the next.
Yeah.
How wavelet transform is correlated with CNN concept in comprehensive sensing of images.
Okay.
I'm sorry,
yeah.
in compressive sensing of images.
Mm-hmm. Yeah, wavelet, it's a mathematical tool. So basically, you can the advantage of wavelet, you can go for more you can decompose your images into different frequency bands or sub bands, like low frequency, high frequency, low high, like vertical details, horizontal details, diagonal details, or approximation details. So again, you can go lifting techniques and other various wavelet techniques. So in deep learning, specifically speaking, as of my understanding, there is no special relation, but you can use wavelet for the pre-processing. But if you are using a machine learning, you can extract different wavelet features, like approximated features, or the 3 or 4 after using the multiple levels, you can take the approximation features, diagonal features, vertical features, horizontal features. You can extract the features in machine learning approaches. But in deep learning, we use already the fixed filters, so you can use it for some kind of pre-processing applications. And compress the sensing, you mean the compression is another study. So you have to concentrate on how to do the image compression. If you mean for the compression, you have to concentrate. That is like a different study, like a data formats, how to make JPEG and similar kind of study. Compression, lossless compression, how to make a more lossless compression. That is another different study. In compressions, if you mean, I don't know exactly what is the question mean, but if you asked something like compression and wavelet relation, of course, JPEG 2000, the basic standard of transform using for the image compression is wavelet. Yeah.
For pre-processing stage, is there any tool that would reduce the image noise?
Oh yeah, that's what we were discussing. So pre-processing, uh, what we were saying here, we don't go for much noise reduction, but we will go for the pre-processing means. Here what I did for some specific applications, segmenting the our tissues, normalizing into MNI space. So the normalizing, usually we will do the registration and alignment matching. We can avoid the mismatching. Then we will going for Gaussian smoothing filter. That Gaussian smoothing filter also, we will get a smooth images after normalized. Similarly, you can use, depends upon the what kind of data you are getting. You can use for any pre-processing. Pre-processing, you can use any imaging. That is an imaging technique. You can use deep learning for pre-processing, or you can use pre-processing for deep learning. Either way you can use.
Shall we give DICAM or Nifty format data set directly to CNN?
Yeah, that's what we were discussing. This NiftyNet, Nifty Torch, these all platforms are designed for directly using the Nifty images, and similarly DLTK, Deep Learning Toolkit. Those like medical imaging specific toolboxes we can use for Nifty and DICOM images, and NiftyNet and Nifty Torch mostly supporting Nifty format images. And I understand Nifty Torch also support DICOM and Nifty images. But when you go for a conventional way, people used to convert it into JPEG, and sometimes we used to train in that format also. We need to, but that is more complex nature. But that is a premature kind of training. But the more accurate is NiftyNet or Nifty Torch or similar kind of any other platform. This is I just given as example here, NiftyNet and Nifty Torch.
Hello, thank you, sir.
Oh, okay, fine.
So thank you for your wonderful session.
Mm-hmm.
Now we'll move to the valedictory, sir.
Okay.
Hello.
Hello.
Respected participants, good morning to all. This is Dr. Arun Shekhar, Assistant Professor, Department of ECE, GMR Institute of Technology, Rajam, Andhra Pradesh. I thank you for participating in this one-day webinar on deep learning in biomedical image data analysis, organized by Department of ECE, GMR Institute of Technology on 3rd July 2020. So I thank the resource person sharing his value content with GMR and a large community on the online platform. Once again, I thank the resource person for accepting our invitation and which made us to utilize this time in an effective manner to upgrade our knowledge and understand biomedical image data analysis. I thank the participants for their enthusiastic participants in this. I hope this webinar will be informative and worthwhile. On this occasion, I thank the management, our principal, Dr. C. V. Prasad, Vice Principal, Dr. Murugadas, for their and for enabling the necessary in conducting this online webinar. I also thank our head of the department, Dr. M. B. Nageshwara sir, constant support and making this online webinar. Finally, I thank my co-coordinator, madam, and the source directly and directly involved in organizing this webinar smoothly and making it a grand. I think nearly 100 plus participants are seeing this to in the YouTube live streaming. I wish you stay home, update to all. I thank the show directly involved in organizing this webinar smoothly and making it a grand. I think nearly 100 plus participants are seeing this to
the YouTube live streaming. I wish you stay home. Appreciate you all.